Copyright Stanford University, all rights reserved.
And then today we have Dr. William Slikker, who I was fortunate enough to meet probably about a year ago when I visited their site. And I was blown away by what their site has to offer, and that actually most academia folks are not aware of this. And so by way of background, Dr. Slikker received his bachelor's degree from UC Santa Barbara. He then went to UC Davis for his PhD degree. He then went to do a 2-year postdoctoral fellowship at the NCTR, but ended up staying there for 37 years. And so he's been the director of the NCTR for the past 7 years. And the NCTR is the only, I guess as I was explaining, it's the only agency, only branch in the FDA agency that does real research. Most of the other branches are more for administrative paperwork, regulatory affairs, whereas the NCTR actually does research, and they have about 600 investigators within the NCTR in Arkansas. So Dr. Slikker has published over 300 papers, mostly in the areas of transplantational pharmacokinetics, developmental neurotoxicology, neuroprotection, systems biology, and risk assessment. He used to be he has held several committee chairmanships, including the Teratology Society, serving as the president, also the American Society of Pharmacology and Experimental Therapeutics. He's currently the chair of the Developmental Pharmacology section. He's also currently the associate editor for neurotoxicology and toxicology sciences, and he was the past president of the Academy of Toxicology Sciences. He's also on the national and international advisory boards for pretty much all the important programs in our country, including the NIH, the National Academy of Science, the World Health Organization, the HESI, the Health and Environmental Science Institutes. And I think, as you are probably aware, over the past year, we brought in several speakers from Big Pharma, and this is a good, a tie for you to kind of understand how this research works from the basic to the translation and to the regulatory affairs. And with that, I'm very happy to have Dr. Slikker here. Thank you, Bill.
As David is getting this up and running, I just want to tell you that I'm very pleased to be here. It's a great group to be able to interact with. We already have some ongoing studies with some of your faculty, including Dr. Wu, but we'd certainly like to have more. One thing that we do have at the NCTR is the opportunity for training. Thank you, David. And we do have at any one time about 70 or 80 postdoctoral fellows at the NCTR working in various laboratories there. And so I encourage you to look at opportunities not only for postdoctoral work, but also opportunities for doing special projects within your PhD. Now, the other thing I wanted to say is I had the opportunity to meet with several professors and with Dean Miner this morning, really enjoy that interaction. And I know I'm going to get the chance to meet with several more of you this afternoon. But I think part of the idea of this whole process is to build partnerships. And that's one thing that the NCTR, the National Center for Toxicological Research, really represents. We build partnerships with universities, also with other government agencies, as well as even industry. We can develop cooperative research and development agreements with industry to develop certain kinds of mechanistic studies that we can do together. Not only do we do this domestically, but we also do this throughout the world. We have a global coalition for regulatory science that really promotes the idea of interaction with other regulatory agencies worldwide to develop regulatory science principles that can be applied to drugs and foods, devices, vet med products, as well as now, of course, the interest in Center for Food Safety as well.
Sorry about that.
Yeah, no problem. So we are still working on the technical aspects here, but getting closer, I think, hopefully.
It was working earlier.
Mm-hmm. We need to do that. So while they're finishing this up, let me say that over the last several years, we've been looking at the effects of anesthetic agents. And anesthetic agents are very critical, important clinical tools. I wouldn't want to think about going into surgery without them. At the same time, I think that over the last 10 or 15 years, there's been some issues developed around the problem of applying them to developing animals and perhaps children. So we're still working on that. And we get a little advertisement for blue jeans. This is good. The point is, so I can breeze through my introductory slides by talking to you about them now, is that we've been noting that there's a increase, of course, in the use of anesthetics because the use of them for children care is really increasing. For example, David, there's David, all right. The idea is that these agents can be very useful for surgeries. Now, the issue is that within the United States, we have between 5 and 6 million children anesthetized on an annual basis. And thank you, David, thank you. No problem. In doing such, there's the opportunity for exposure to the very young infants. As many as 1.5 million anesthetics or anesthetic episodes are performed each year on children that are less than 2 years of age. Let's see if this thing will actually advance. Aha, we will. So now what we're interested in is trying to understand a way to use these anesthetics safely. And I think that one of the things we've been focusing on is the data that we generated in animal models. And I'll go in later some of our attempts to move this into the clinical setting, which is a more difficult task. One of the things I want to sort of challenge some of you with, especially those in the audience that use anesthetics, either as anesthesiologists or as surgeons, as to the best way to arrange a clinical trial using anesthetic agents. I think you can imagine right up front that is a very difficult thing to do because what is your control arm? We can do that in animal models, but it's difficult to do in humans, but especially human children. So it's a difficult challenge. I want you to be thinking about that as we're going through these slides, and hopefully you can help me figure out a way in which we can move this area forward, especially when it comes to application to children. Now, one of the things that happened in 1999 is that a group at Washington University in St. Louis, John Olney's group, identified that cells seemed to be dying in the central nervous system of rodents that were exposed to ketamine, which is an NMDA antagonist type anesthetic, and other agents that affected the NMDA pathway. And this was kind of a shock, a wake-up call, because before that, it really wasn't obvious that there was issues with the use of anesthetics in developing animals. Later, we were able to confirm those findings. Andy Scallett's one of my colleagues that worked with us at NCTR. And we were able to do dose-response studies to identify this issue and to replicate those findings. And then more recently, we moved this model to the non-human primate to make sure that it was something that worked across species. And indeed, I'll show you that data would suggest that the non-human primate is also sensitive only during certain developmental periods of time. And then work really moved forward to understand something about the reversibility and longevity of this effect in animal models, and my colleague Merle Paul at NCTR did studies that were exposed the same way to ketamine. These animals were exposed on day 5, but follow them using behavioral assessment tools. I'll show you some of that data. And indeed, even after 3 years, there still seems to be a lingering deficit in these animals after a single exposure to anesthetic ketamine in this case. And then more recently, there's been some data coming from human studies, although the critical prospective human trials have not been done yet. We're trying to move in that direction, but some early data coming from Randy Flick's lab and others suggested there may be an issue with some developmental profiling in humans that were exposed during early childhood and infancy to agents, especially multiple exposures to anesthetics, and then even more recently in that, this cohort that suggested there may be issues even with the single exposure to an anesthetic agent. So we'll get into that in more detail, but I just wanted to mention, of course, as most of you know in this room, that the two different ways in which most of the traditional anesthetics work are either through the NMDA receptor, and these are the GABA receptors, and we'll talk about this in more detail, but these pathways, of course, are very important biological pathways in normal development and in learning and memory, and so one can understand is by modulating them, you can also do good as well as possibly do harm. So these are some of the agents we'll be looking at. Ketamine is not used that frequently, if at all, in adults, but it is used in children, and it depends on the medical center and a host of anesthesiologists, but it's a very quick onset, quick offset kind of anesthetic, and one in which certainly there's very little effect on the cardiovascular system. Nitrous oxide is old standby, oftentimes mixed with other agents, and then your GABA agonist, propofol, is one that we'll talk a little bit about. And then, of course, the sort of workhorses of anesthesiology, either the isoflurane nitrous combination or sevoflurane, which is most popular now. These are both gaseous anesthetics that are quite popular. We'll look at a little bit of data from all those. I won't spend very much time on sevoflurane because the data has just in the publication mode right now, but we'll spend more time on the isoflurane data, and the data sets are very similar as it turns out. So, just the cartoon of the NMDA receptor, just to give you an idea about the importance of this channel for calcium flux, and the idea that it's made up of 4 subunits. These subunits contain always the NR1 subunit, but can contain a variety of other subunits. And these either can be totally NR1 or it can be a subgroup of several of these units to make up the functional NMDA receptor. But we've used this advantage of NR1 being a major component in some of our subsequent data, which I'll show you. Now, this is some of the initial dose response curves that we did. In this case, we're dosing rodents on postnatal day 7. It had been shown by others that postnatal day 7 is probably the most sensitive time period for rodents, although you can get effects in day 3 of age as well as out to day 14, but not beyond that as far as these particular anesthetics. What we're looking at here is an amount of anesthetic that it takes to anesthetize a rodent with ketamine, and this is a lot more drug than it would take to anesthetize a monkey or a human. That's just the way it works. If you're going to get a certain anesthetic plane, you've got to use these kinds of doses of right around either 10 or 20 milligram per kilogram, given every 2 hours because of a short half-life, for up to 12 hours total. And when you do that, you can see a dramatic increase in the fluorojade staining of neurons from the brain regions of this rodent population. And it's also pretty obvious that there are certain areas of the brain that are more affected than others. It looks like the frontal cortex, at least in terms of our data, is one of the more sensitive areas, but you also see significant effects in the striatum, the hippocampus, as well as the amygdala. So it seems to be somewhat of a general effect in causing the cell death. Those that maybe aren't familiar with fluorojade, it actually was invented in NCTR by Larry Schmood, and it's one of those agents that helps identify dead and dying neurons. But we've also done other approaches, such as a variety of ones, including silverstain, that I'll show you later. So this gave us the first idea of something about the dose response, a favorite thing for pharmacologists to do, and to look at where the issues were with this particular agent in the rodent. We also did some distribution studies. Pharmacologists like to do this. Where is the drug and when is it there? And what you see here, either in the plasma, shown in red, or the brain level, shown in green, is that after this series of injections over 12 hours, and looking almost immediately after the last dose, you can see substantial levels in the plasma and about half that or so in the brain tissue. But what you see is very short half-life, and so by 2, especially out of 4 hours, you virtually have eliminated most of your ketamine in the tissue or in the blood. And what's been interesting about this is that if you look at an ELISA cell death assay, what you see is it doesn't sort of signify cell death until you get out to 6 or even 12 or 18 hours. And so there is a delay, and this was very interesting to us. Why is this happening after the drug is gone, then you get your cell death? And part of it could be the normal cascade of events that has to occur, but we also think something else is involved in this as well. And one of those things that we're interested in is actually the NR1 subunit of the NMDA receptor. Remember, I mentioned that was an essential part of that receptor. If you measure its messenger RNA, you can see that after ketamine exposure, the levels of that message go up. And right along with that, you can see the distribution after the ketamine exposure compared to the saline, which is shown here. So we think that there's something interesting about the effects of ketamine and the increase of the levels of that particular receptor which it interacts with. And you can also look at this a variety of ways. We used microarrays here, as well as a fold change and quantitative PCR. And you can see that indeed the NR1 subunit goes up, as well as the 2A, perhaps the 2C, but not necessarily the other ones. And so it does seem to hold that not only the message goes up, but the actual protein goes up for this particular receptor. So we feel this is something that's related to the activity of ketamine. And indeed, you can make a simple model. This is a working model that we published now several years ago. But we realize that calcium is an integral part of the importance of how the brain functions, and that these NMDA receptors are the ones that bring calcium into the cell. And normal, voltage-dependent activation NMDA receptor by glutamate will open up these things and let calcium flow in. Of course, if you have ketamine on board, it's a non-competitive NMDA receptor antagonist that will block the entry of calcium into the cell, and so you get very little in there. And then if you release the ketamine and it goes away, one of our ideas is that based on the potential upregulation of the NR1 subunit and other subunits of the receptor, you may actually have more receptors there than before. And with those receptors no longer being blocked by ketamine because it's left the system, then you have this onset of calcium influx, which, as you know, calcium can be a very good thing and important to normal physiological functions, but it also, with larger amounts, can lead to necrosis or apoptosis. So one of the things that we've been looking at here is how this actually affects the mitochondria. And one of the things, and I'll show you these in tandem, is ability to look right at the subset of neurons in the nervous system of a rat that was anesthetized with ketamine, and then following that animal 24 hours later, slicing the brain tissue. And what you're looking at here is actually a Gaton 3 kind of approach where we're looking at 50 nanometer sections, over 400 of them sliced through this one small region of the brain showing a neuron and its associated nucleus. And what you're seeing here then is both the control animals and the treated animals and how this affects mitochondrial function. We think mitochondria are some of the point issues with this, and you can color the mitochondria in different colors so you can see them and look at them differentially. And you can compare what we see here in the control animal to what we see in the treated animal. And one thing that's obvious is that these mitochondria are much more rounded and larger in diameter and size than these in the control. And indeed, if you go in and do quantitative measurements, you'll have about a 40% increase in size of mitochondria. I want to give credit to Tricia and also Angel Paredes, who developed this data set. And she was able to take home a prize from the FASEB's BioArt arena with this particular film. So it does, though, I think, signify the importance of the mitochondria, and I'll give you more details about that later in terms of the actual influence. And it is kind of interesting to be able to visualize mitochondria at this level so you actually know what's going on. Now, let me turn to an area that I know is very important to the Stanford campus. It's like bringing coals to Newcastle, stem cells. But for us, it's important because we're interested in the fact that you can have these stem cells develop into various kinds of cells important in the nervous system, including the neurons. So we use this approach using the rat embryonic stem cells. Of course, as all of you know, you can have them days in vitro divide and make really nice pictures here of both nestin and EDU labeled cells. So we know that they're actually developing neurons. The other thing is that then when you combine that with exposure, in this case to anesthetics that are used in children, ketamine we've already talked about. Other one that you know about for a variety of reasons is propofol, another short onset agent that interacts more with the GABA-A system as a receptor agonist. And both of these are certainly used in the human setting, but in animal models, they do both produce some problems in the in vivo setting. So what we want to do is understand what they do with stem cells used as a model for testing various agents. And I know here, stem cells are important for a variety of reasons, including the idea of transplantation and therapies, which is really a great use for them. But in our world, it's really important also to have models that you can use for assessment of agents, including agents that are regulated by the FDA. So what you see here is the embryonic rodent stem cells collected from the nervous system. And you can see that they're collected on day 16, and the growth media is specified here. Changed every 3 days out to days in vitro, day 8. and then you can expose them to anesthetic agents, either 3, 4, 5 hours, up to 24 hours, and look at a variety of endpoints which are shown here. I won't go into all these experiments; they've all been published, but the idea is just to understand how we're using these for our particular use with the anesthetic agents. What we found is that there was very limited effects until you got to large concentrations of propofol. Although you can see here in the MTT assay, giving you some idea about mitochondrial function, that even as low as 50 micromolar, which is in the range of what you would see in the clinical use of this agent, was showing some effects. So even cells that were not differentiated, but were just proliferating, did show some effects. And indeed, you can see that here, where you're looking at cells during proliferation, 24 hours exposure to propofol at 50 micromolars for 24 hours. You can see the decrease in EDU positive nuclei with the propofol compared to control. But you can also see, and I'll come back to this again later too, the use of an agent that we think helps stabilizes mitochondria and blocks oxidative stress, acetylcarnitine. At 10 micromolar concentration, you can see you can protect against this effect of propofol, which is rather interesting and suggests that there may be an oxidative stress type mechanism here. Now, the other things you can do with these cells, of course, is take them on now from where they are just proliferating all the way through differentiation. And you can do that by going through these transition media to a differentiation media, and then you can expose them after they've been differentiated. And again, 24-hour exposure to anesthetic agents. And there, then you can start to see the GABA-A receptor. The GABA-A receptor was not available when the cells were differentiated, but now it is there when in the differentiated cells, and you can see and use that to your advantage. And what we did find is that with propofol, it would affect cells just in proliferation phase. It also affects cells that were differentiated. So it affected them fairly much evenly, but both of them were adversely affected. Now let's move to our friend ketamine at 10 micromolar, which is sort of your equivalent of a clinical type exposure in terms of a rodent, that is. 24-hour exposure, and what you see is cells that are just in proliferation, that there's no effect, okay? There's no difference between control and treated. And what this indicates right away is that you have a different response based on where your cells are in the maturational process. This is really important to keep in mind because you might really have to understand where your cells are in order to use them appropriately. However, if you look at this, and this just emphasizes that fact that ketamine produces no change of control, whereas propofol really increases the number of tunnel-positive cells going to apoptosis. Now, if you take these same cells and now grow them out into the different kinds of differentiation media and have them differentiated at this point in time, then you get a different response from ketamine. And indeed, one thing we're able to show is that ketamine exposure would increase the expression ratio of the NR1 subunit. Again, this is the increase in the NMDA receptor complex increasing in this in vitro setting in rat stem cells in culture that have gone through differentiation. And certainly you can see it here as well as the NR1 subunit going up. Now you can also look at functional effects, and this is really important about cells in culture that can be very handy here. We're actually looking at changes in fluoro2 loaded neurons, looking at calcium flux. And this is in a control neuron. You can see that NMDA would produce a spike, which you would expect, because that's interacting with the receptor. If you add EGTA to that, no response, but you would expect that. Glutamate causes an increased response, and of course, KCl eliminates everything. You get a dramatic response. Now, if you look at animals, or cells from animals, cells that have been treated with ketamine in vitro, then you get a whole different kind of thing. You get an exaggerated NMDA response, an exaggerated glutamate response, and when you quantify that, you can see you get about a 40% increase in calcium influx and loading in those cells after treatment with ketamine, those that have gone through differentiation. So this gives you a functional output and also really focuses again on that mitochondrial function and transfer of calcium into the cell through the NMDA receptor complex. And just to sort of summarize in, you can see that looking at Bmax, one of those pathways to apoptosis, that if you look in growth media, only the propofol produced results that were different from control. When you look at differentiated media, then you get propofol produces the response, and so does ketamine after it's gone through the differentiation. So again, you got to know where your cells are. And that's sort of summarized here, that understanding, anesthetics may be useful to use stem cells, especially since now we're moving to human stem cells where you can get a closer comparison for translation. but also you need to know what developmental stage your cells are in and make that a very reproducible, standardized thing. And then under these conditions, you can probably get data that certainly seem to reflect the in vivo data that we have from ProProFlow, not only from our lab, but from other labs as well. So let me turn now to really trying to nail this down in terms of where are the sensitive periods for exposure and how does it translate between species. And so what we were able to do is that we had data sets from the rat, us and several other labs across the country. We knew that the rats were sensitive from somewhere around birth up to about 14 days, with peak sensitivity at day 7. Now the monkeys, at this point in time, we didn't have any data, but we guessed based on the idea, where is the synaptic development of that brain? How rapidly is it going through synaptogenesis and differentiating? And what we surmised, it would be somewhere around mid-pregnancy up to about 2 months of age. And then you would have a period where they would not be sensitive. These effects down here are the other effects of ketamine, for example, which many of you know causes vascularization given to the adult animal. That's a whole different phenomena. So that the period for humans then, and this is just a guesstimation based on their brain development, will be somewhere around the last third of pregnancy up to maybe 2 or 3 years. And this is a very gross speculation right now because we still don't have the data to fill that human one in. But we do have data for the rhesus monkey, and I'll show you that next. So what we did is a number of preliminary experiments to make sure that we could do the technique. Then we looked at determination of the sensitive stage by exposing during different critical periods of development. We also looked at the duration of exposure, whether you went 24 hours, 9 hours, 3 hours of exposure to anesthetic agent. And we also looked at reversibility using behavioral assessment tools. And then we also did some bioimaging as well as some omics determinations. So all these together generated a data set that's relatively complete for the non-human primate, the rhesus monkey in this case. We also, just like you would do in an OR for humans, we measured all these things, okay? And actually our OR for our monkey surgeries looks kind of similar to what you might see in the human situation. And we also looked at plasma levels of ketamine, as well as the blood gases, body temperature, et cetera, et cetera. So in doing so, we can actually look at the levels of levels of ketamine in plasma in the developing monkey. And you can do this in the various stages that we evaluated. We evaluated the gestational day 122 animal. Now, the rhesus monkey has term at 165 days, plus or minus 5 days. So 122 days of gestation is somewhere around 3/4 term. We also looked at postnatal day 5 animals, and all these were 24-hour exposures to ketamine. And we also looked at the postnatal day 35 animals, which you see here. And then just to try to get some idea about duration and how that was important, we looked at 3 hours of exposure. So what you can see from these plasma levels is that they're about 3 to 5 times higher than what you see in the human. And that's because it just takes more ketamine to anesthetize a monkey than it does a human. Less than it takes to a rat, but more than a human. These animals were followed by a veterinarian, a clinical veterinarian. We maintained anesthetic plane that one would use for surgery. And we did this by a variety of measurements, including the physiological measurements, as well as using various other kinds of standard controls to make sure the animals were anesthetized at a proper plane. So in doing so, this is what it takes to anesthetize the animal. This is a 3-hour exposure. You can see it's right on top of the longer exposures. What's interesting is this higher level here is required to anesthetize an animal that's postnatal day 35, whereas these lower levels are used to anesthetize the pregnant mom because it was 122 days of gestation, and the 5-day-old monkey. And then, of course, after the animals are delivered and you look at them 6 to 12 hours later, you can see the levels drop way down, as you would expect. So this gives you an idea where we are pharmacologically compared to the human situation, and also that the postnatal day 35 exposure to anesthetize an animal postnatal day 35 is a little bit more necessary of ketamine than at day 5. Now, just to give you one snapshot of some of the things that we measured. We looked at percentage of hemoglobin, which is saturated, a typical thing you'd wear on your finger. Here we put it on the tail of the animal. But in any case, you can see that there was really no difference between the O2 saturation in the pregnant females that were anesthetized or not anesthetized, the postnatal day 5 animals, or the postnatal day 35 animals. Just one of those many endpoints that were the same. However, their brains did not look the same. Okay, we collected tissue, stained them, in this case, with silver stain, which is your classic method to look at cells that are dead or dying. And if you compare your control animals with 24 hours of control conditions compared to 24 hours of ketamine anesthesia, in this case, you're looking a day later, you can see you have an increase in silver stain, especially in layer 2 of the cortex. The 3-hour exposure to ketamine as compared to the 24-hour did not produce any significant effects. So you can quantitate all this, put it into a nice bar graph, and you can use markers such as caspase-3. You can look at silver stain, which is your classic approach, and you can also use fluoro-J, which gives you the same kind of data. But what you see is that 24-hour exposure to ketamine in the pregnant animal, 3/4 term, when you looked at the fetus, you did have increased cell death with all these techniques that we used. Same way with the postnatal day 5 monkey, not so with the postnatal day 35 monkey. So somewhere between day 5 and day 35, the animal stopped being sensitive to this effect of anesthetic. This is very important to try to think about how to design a clinical study and also to protect the clinical population. And we also found that if you do a shorter exposure, we did not see any effects of increased cell death. So that was also encouraging. So somewhere between 3 hours of exposure and 24 hours of exposure to anesthetic, the animals changed from being sort of non-sensitive to sensitive. What we did then is also a 9-hour study, which showed to be positive, and others have done now 5-hour exposure, at least with gaseous anesthetics, as well as our work at 5 and 8 hours, and they're sensitive there. So somewhere 3 or under, they seem to lose sensitivity, which is a good thing because that's going to take care of about 80% of the anesthetized children that are performed each year. But you still have that 20%, some of which are undergoing cardiovascular surgery and/or nervous system surgery that are going to be exposed longer, and that puts you squarely in this area here with that 9 hours, 5 hours, and 24 hours of exposure. Now, let's look at something about how long these effects last and do they have functional consequences. One could argue that a few dead cells in the nervous system, especially in early development, it's going to clear up, they're going to phagocytize, the signal goes away, the cells that are there, that are functional, continue to function well, and everything is just fine. No real problem with losing a few cells in the nervous system, even though it's around 10 to 40%, depending on what paper you read. So is there a functional consequence of that cell death? That's one thing we wanted to look at. So at the NCTR, over the last 25 years, we developed this operant test battery that allows you to assess cognitive function in rodents, primates, and humans, and it actually uses the same equipment in all cases, so you can compare across 3 different species. And we look at learning, motivation, color, and position discrimination, as well as short-term memory with these particular approaches. What we found, and this is early postnatal exposure to ketamine, does it produce long-lasting cognitive deficits in the rhesus monkey? So we did a 24-hour exposure. Either they were exposed on postnatal day 5 or postnatal day 6. We weaned them out to 6 months, and they started training around 7 months of age to do these operant skills. And then we monitored them for 2 years. Currently, we're up to 4 or 5 years. This data is older now, and I'll show you what effects we found. One thing we need to realize is that we're looking at different kinds of cognitive function, either learning, motivation, position description, memory, all within the same battery of tests. Every 2 days, the animals repeat 5 days a week. We get data, hundreds, if not thousands, maybe now millions of data points from these animals that we compare against where they were before and how they transition and compare between groups. And this is just an example of how we do this in children, where instead of working for banana-flavored pellets, which the monkeys like, children aren't too fond of those, but they do work for nickels. And so we call this our nickel game, and it's a series of press plates up here that are backlit and levers here and indicator lights here. And by working this system, they can garner nickels, and we can do the same kinds of tests in children as in animals. This is just an example here of full-scale IQ with children that obviously have a variety of IQ levels. You can look at accuracy in our learning task, which is one of the 5 different tasks that we're looking at in this operant test battery, and you can see a nice positive correlation. What's interesting, if you look at this with motivation, there's no difference between the kids with the different IQs, but there is in their ability to learn. Now, what are effects on our monkeys that were exposed for 24 hours to a single bout of ketamine anesthesia, and you follow them over the next several years? They started training at 7 months of age, and you can see as we train these animals, you can't just show them a videotape as you do the children and say, this is how you play the game, now go play it. What you have to do with animals is shape their behavior and get them to start playing these games, first by taping pellets onto the levers, and then eventually they figure out that if they press a lever, they get a food pellet. That works very quickly. And then you start training them on these other components. This is a learning task. It takes a while to learn that. The animals between the treated and control were pretty much the same with a one-task progressive ratio added there. They started to learn that. This is your position definitive description kind of response. They learned that. But when we added the delayed matching the sample, which is a short-term memory task, you can see the animals began to separate in terms of the treated versus the control. And as you follow this up and did the complete array of delayed matching the sample, which gives you variable times before you have to match the sample, you can see that the treated animals do not match up. In fact, you can look at components of this, and in this learning task, which is our incremental repeated acquisition task, the learning task, you see a difference that separates the control from the treated out many months. And in fact, some of these changes are still there 3 or 4 years later. So whatever the effect was, it is a lasting effect, and it does match the same animals that showed those loss of cell types in the various brain regions. So let me move away from that and move to the area that is actually more exciting for us. Identifying this issue is one thing, trying to ameliorate it is something else. And so we get into some more of the neuroprotection area. One of those things is to look at antioxidants, because we felt from our earlier data, both in stem cells and other approaches, that you were getting oxidative stress. And so we're looking at different in vitro experiments and in vivo experiments that have used a variety of agents, including the L-carnitine, which I mentioned, and I'll show you some more data on that, that have had a tendency to ameliorate these effects. I'm sorry about that. Okay, so we still got some more time here, folks, so don't leave yet. Okay, so what we have here are classical agents that have been used to block this effect, and all those are published by the people shown. I'm going to show you a little bit of data about the L-carnitine effect. These are just some of the agents that have been used. Lithium is an interesting one. It seems a single dose of this will protect against this effect. So were melatonin, and the one that we've studied the most so far is this L-carnitine or acetyl L-carnitine, which gets to the target site a bit better than L-carnitine. But many of them have been tried. Just a quick set of experiments using the 7-day-old rat pup model, which is one of your most characterized models. In this case, we're using the gaseous anesthetic isoflurane at 0.55. For those that are anesthesiologists in the room, that's kind of low. You could be using 1 to 1.5, but it is on the low end because we coupled it with 75% nitrous oxide, a classical kind of anesthetic protocol. And we did it with and without L-carnitine as a neuroprotective agent. And what you see is that if you look at nitrous oxide or isoflurane by themselves, that they don't produce any effects even out to 6 hours of anesthesia. So, but if you combine them, then you start to see effects, especially out here at the 6 and 8 hours. And you can see here in the bar graph when you quantify this that these animals are sensitive. What is especially important about this is, does the L-carnitine protect? And so here we started looking at, well, you can also look at silvergrains as well as fluorojade or caspase-3. All of them tell you the same kind of story. You can definitely tell that these cells are not happy here when you have the 6 hours of nitrous oxide and isoflurane together. But here, what was interesting part is that if you start adding L-carnitine to this, and you give this in 2 divided doses over the period of anesthesia, you can see a dose-related decrease in the toxicity. It doesn't get quite back to control values, but it cuts it by more than half when you add the L-carnitine. And so we feel like this is at least leading in a direction that might provide some protection, and it also talks a little bit about the possible pathway to toxicity, which seems to be related to oxidative stress. So now let me turn finally to our bioimaging studies. There's a lot of imaging done on this campus. It's a very important preclinical tool, we believe. And so to advance that, we've used both the 7 Tesla MRI, but I'm going to talk mainly about the micropet data here, which a monkey will easily fit into. What we've done here is first looked at a very simple compound in animals that were exposed to the traditional paradigm of ketamine given multiple doses in postnatal day 7. This is the 6 subcutaneous injections of 20 milligram per kilogram ketamine received every 2 hours over a total of 12 hours. And this has been published with an Exon 18 label. I mean, how this works is very simply, is that when your plasma membrane goes through apoptosis, it externalizes the phosphatidylserine, and in doing such, then it makes a nice place for the annexin to bind to. And in doing so, it's going to keep that annexin around longer. So if you tag it with F18, you can follow it, and it'll be around longer in the body. And here you can also focus the micropet, and we looked at the sort of the more the cortical region of the rat brain and postnatal day 7 or 8. And so what you're able to see then is that over time in the control animals that weren't exposed to anesthesia, that the signal goes away quite rapidly because it's a normal half-life of F18 Nexin 5. But in those animals that were treated with ketamine, you can see the signal stayed much longer. And indeed, when you do the quantitation of this, you can see quite a difference between the ketamine-exposed animals here in blue compared to the normal release of this F18 labeled Nexin 5 in black. And also, the idea here is that these animals are alive and well. We can use them again. We can study them later. We can study them in behavioral studies. We can look at their neurochemistry at some time later, histology, whatever. But we have evidence right here. These animals are different. And we believe it's because the Nexin 5 hangs around because cells are going through apoptosis, which is what the neurohistology also tells us in these same kinds of preparations. And so the other one that we developed is this one called FEPA. We found the Nexin 5, as many people have over the years, didn't really transfer very well across the non-human primate brain. The blood-brain barrier kept it out. So we went to FEPA, which is another kind of label, and I'm sure some of you here have used it. Certainly was developed in part by a number of labs, including Thomas Ghilarti at Hopkins at the time. But it interacts with the peripheral benzodiazepine receptor, also a transport molecule, they're one and the same. And in doing such, it's thought to signify damage and inflammation that occurs when that particular compound, whatever compound it is insulting the brain. FEPA levels tend to stay around much longer because they're signifying this inflammation that occurs. And how that we think that works is that the F18 labeled FEPA binds with cells, especially on the membrane of the mitochondria, and in binding to this translocator protein, which is upregulated when cells are going through this effect of oxidative stress and immune dysfunction, then you can use them as an indicator of mitochondrial dysfunction and increase in oxidative stress. So in doing such, you can see right away that in animals that were dosed with ketamine on postnatal day 7, and then 7 days later after the exposure, we looked at the amount of FEPA that was still around, and you can see that there's quite a difference when you inject the animals with the FEPA. It stays a lot longer, those animals that were exposed to ketamine a week earlier as compared to controls. And as we expected, on day 14 after exposure, day 21 of age, you get a lot more variance in your treated animals because some of them are showing effects and some of them are not. You would expect this because cells go through phagocytosis and get cleaned up, no longer have the signal anymore. And when you go here to 21 days after exposure, you can see them coming right together in the same way at day 35. So one of the advantages of using imaging is that you can follow the same animals over a time course. And doing such, you can tell that this signal goes away over a period of time. You would expect that because these cells don't hang around forever. They have to be phagocytized and eliminated, and then your FEPA signal goes away because the inflammation goes away. So now, does this work in primates? That was the big question we had. And so we did our experiment in primates, non-human primates, where we're looking at nitrous oxide at 70% and isoflurane at 1%. And we did this also in animals that were exposed either individually isoflurane, nitrous oxide, or the combination. And when you look at all the physiological parameters, which are very important to keep in mind, you have to remember that 5, 6 years ago, when we first brought this up to audiences of anesthesiologists, they go, hey, you guys are obviously doing something wrong. It's something wrong with your physiology of your animals. They're not getting enough glucose. They're hypoxic. Well, that's why we did all these measurements. Is it still there when you do all the physiology measurements? And indeed, you'll see it's still there. So here we got caspase-3 aminostaining in frontal cortex of the primate exposed to nitrous oxide and isoflurane. And this is for 8 hours of exposure. Here's your fluorojade. Isn't that beautiful? Fluorojade really works, folks. It has over 800 citations now. It's such a popular technique. Anyway, when you put this all together and look at the effects of nitrous oxide and isoflurane, with that 1% isoflurane, 70% nitrous oxide for 8 hours, you did it in the postnatal day 5 or 6 day old monkey, and you follow them, in this case, over a period of time, in this case, just one day. What you see is these effects after histology measurements. This is fluorojade again and caspase-3. So there is an effect of these agents, and you can also pick it up with imaging. And that's what we're showing here is the PEPA. And these same different animals, because those animals were sacked, obviously. These animals were treated the same way, but we follow them over time. And you can see here in the temporal lobe that you definitely have an increase in PEPA half-life with this particular exposure to anesthetic agent. And you can also see this in the temporal lobe. And you can also do the acetylcarnitine. Remember I mentioned that as an antioxidant and mitochondrial protectant. And what you see is that when you combine the two here, that the levels come right down pretty much to control values compared to, this case, isoflurane nitrous oxide alone. So here's an anesthetic that's used routinely in the OR for many different patients, and including children. And yet we know this particular effect is there now, at least in the non-human primate. We don't know what happens in the child. So here we go with the advantages. Again, you guys know all these advantages, but it does allow you to do many different things when you're using imaging as a preclinical tool. Now, let me just get down and summarize here. Over the years, our lab and others have developed various kind of pathways that may be activated in this case. We certainly feel like this GABA A receptor as well as the NMDA receptor are critical pieces of this puzzle, and that the anesthetic agents have a tendency to make them malfunction to some extent. And you may be aware of this, that in development, the GABA A receptor, which in the adult is normally inhibitory, it is excitatory during development. So it has a different role, which may add to the issue here. And the NMDA receptor, we've already talked about why we think it produces issues by upregulating the NR1 and other subunits. So here you have the effect on the mitochondria we think is a central part of this, and I showed that video earlier showing the effects of mitochondrial swelling. We think that mitochondria play a major role. We certainly know that this whole cascade to apoptosis is there and stimulated by this effect. We also know that you can use various studies looking at NF-kappa B. If you block the translocation of NF-kappa B, you can block this effect and other kinds of mechanistic things. And now we've turned our attention not only to the imaging that I've showed you, but also to other potential biomarkers. That is, what would happen if you looked at phospholipid integrity in terms of the availability of lipids in the blood, and we're doing that now, and also looking at the cytokines. And we do have some preliminary evidence that some of those select few are elevated or decreased based on the exposure to anesthetic agents. So we're looking for minimally invasive markers of toxicity that could signify if you have an issue with the anesthetic agent. And these can be translated to the human situation, which is one thing that's very critical. Now, let me just summarize by saying that we're not alone in doing this work. There's been over 70 rat studies, and although we're probably responsible for about a third of those, many other labs have replicated this across the US and Europe, and all these different kinds of endpoints have been evaluated. In the non-human primate, we've probably done about half of these or more studies, but other groups at WashU and University of Oregon have also done these in the non-human primate, and all these things have been looked at, and they're activated by these particular sets of anesthetic agents. So it's not just our lab, and it's not just one endpoint. There is an issue here in animal models. Now, conclusions, and I'll go through these quickly because the time is late, but first of all, this phenomena has been observed in all the species studied so far, all the way from roundworms, zebrafish, up to pigs, non-human primates, mice, rats, guinea pigs, etc. And there seems to be some sort of a threshold kind of effect where you have to have exposure for a certain duration before this effect occurs, and during a certain sensitive time of development, which at least in the monkey seems to be somewhere around midterm out to about 5 to 35 days of age, in that range. How that translates to the human is something that's a question. We think that certainly these receptor systems are involved in that process, and that almost every agent that's produced this effect, except for xenon, hasn't been shown to be effective in producing this detrimental effect, but it is a fairly effective anesthetic, so it may be one direction to go. Also, we find that the animals seem to be most sensitive during rapid synaptogenesis, and that there are certainly functional effects, and it depends on which brain area you're looking at the time. Now, these functional observations are important to cognitive function and general intelligence, but you have to be able to measure them correctly and quantitatively. And we often worry about the translation of these well-tuned animal experiments to the human population, where we may not have the power to evaluate the subjects as thoroughly. So we're actually using our test battery in several ongoing studies in university hospitals and other places where they're evaluating children, and therefore you get a species comparison of the data after exposure to anesthetics. We worry about the effects and how long, how long lasting the effects are, but at least in our animal models, it's for years. We also understand the importance of, that cell death may not always accompany these exposures, so we're still understanding some of the vagaries of that area. And that there are several approaches that seem to ameliorate the effects, including using other kinds of anesthetics that are not NMDA or GABA related, or perhaps agents that block the effect. Then it also appears that there may be a sex difference based on some early data, but that certainly needs to be clarified. Also the interaction with surgery. There's one thought, well, surgery may modify the outcome, because obviously in humans, you're going to be doing surgery if you're anesthetizing an individual. And in that particular case, what little data there's out there in rodents suggests that it may actually amplify the problem and not protect against it. Also, the idea of when you expose is very important. And drug combinations, that is, if you're using several anesthetic agents, it seems that it necessarily may not make it better, it may make it worse. And especially interaction with caffeine. This is something that John Olney's group at WashU just recently showed, that interaction of caffeine, which is oftentimes used in the pediatric wards to increase cardiovascular heart rate and to increase breathing, it may actually be an added issue when you add anesthetics with it. Okay, and then finally, so what questions remain? I challenged you early on to think about how to design a clinical trial that would really help us move this area forward. It's a very difficult thing to do, but I certainly welcome your input. And we need to worry about things like in the human, what is the threshold for exposure? How long can you expose to anesthetic and still not have a problem? And is there a problem if you expose for a longer period of time? An open question. Also, the period of sensitivity. We kind of know where it is in the rhesus monkey, but how does that translate to human? Are women who are exposed to anesthetics during mid and late pregnancy also able to have this effect on their fetus? We don't know that yet. And then, of course, how you translate what we do know in the animal models in terms of developmental profile, to the human situation. So I just want to end by saying that in order to try to address some of these issues and translate this kind of information to the clinical setting, FDA, along with the International Anesthesiology Research Society, have started a public-private partnership called SmartTots, and we use it to not only talk about this effect and to inform not only parents, but also inform physicians very carefully about where we are with these sets of studies, and also hopefully to raise funding so we can do these clinical trials in earnest. We think it's going to take about 20+ million to do these studies well in children, even though we haven't come up with the best design at this point in time. That's where we need your input. And then finally, talk about acknowledgments. Certainly the NCTR staff, Chen Wang, has done a lot of the studies in vitro as well as in vivo. Merle Paul has done the behavioral studies. Zhang Shuang has done a lot of the imaging studies. Fang Liu has done a lot of the in vitro stem cell work, along with Shilong Liu, who's done a lot of the PET imaging studies. And my colleagues and other centers of FDA, partners, National Institute of Child Health and Development, the National Toxicology Program, other parts of FDA, including the NCTR, funded this work. So with that, I'll close and I'd be happy to answer any questions you may have. Yeah. Yeah. Well, it's obvious that you noted very well what we've done with ketamine and an NMDA receptor complex. We're not sure how that will translate to the GABA system, but it certainly is something to consider. It's interesting in that I just reviewed a paper looking at NMDA turnover and dynamics, suggesting that if you withdraw alcohol-dependent individuals at a certain rate, you can decrease the subsequent toxicity. It's just a model, but an interesting one that kind of flows into your idea. Is it something that can be done in such a way, not having this immediate withdrawal from the anesthetic? Interesting point. It doesn't really make the surgeon or anesthesiologist happy, because the first thing I want to do is get out of there and get this patient back breathing again and leave the scene because the patient's fine. So it doesn't really fit into their schedule, but it is something to consider. I don't know whether it will work. We could start with animal models to see whether a slow withdrawal would prevent this effect. Interesting point. Yeah. 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 We have really not addressed the benzodiazepine question, but certainly it is an agent that's been used in the anesthesiology world. The group that has addressed this the most is Vesna Tordovich at University of Virginia, and she does have some data on this suggesting there may be issues, but I'll have to say that it hasn't been the main focus of most of the research to date. There seems to be some interaction, but as you know, it's oftentimes given in conjunction with other anesthetics as a pre-dose kind of thing, or maybe subsequently. And so I would say right now, until we develop faster models, maybe cell culture models, we really can't address that issue thoroughly. I think it would be something that would be worthy of consideration because they are used in large amounts, you're right.
I comment on, since most of us try to develop new that treats and improve, and in your case, you're trying to identify. I guess I find it a little bit interesting that if you have all these toxicities and you're the FDA person, why do people still use it? And is this enough? And is there any other alternative to it that people ought to be using? That there are quite a lot of toxicities. Let's just get rid of these. Yeah. Work on something that less.
Yeah, well, what we've looked at here are the primary anesthetics that are used clinically today. And there are others that are being considered. And maybe some of them who don't use these pathways of either GABA-A or NMDA would be useful. It's still unknown whether or not they're safe and effective, but they are being evaluated for this very reason you speak of. But you're in a situation that's a very important one. No one wants to do surgery without anesthesia, so what are you going to do in the meantime? And that's why we're rapidly trying to get to an answer and rapidly trying to get funds to really look at these in the human condition to see whether or not this phenomena translates to the human. You're right, if one was developing a new drug and saw these animal data, do you think the drug company would develop it? So we're in a situation where we're having a potential influence on clinical practice, and this is a long-term investment.
Copyright Stanford University, all rights reserved.